So let's start a new session right away. And Pankaj, I think, already well introduced himself to the audience through his many comments and questions and contributions. So Pankaj, please share your screen and shoot.
I assume everyone can see my screen.
Looks good.
Yes.
All right, let me start my timer. All right, so thank you for inviting me. So I'm going to tell you about some thoughts I have about thinking about interactions and collective modes and how we can use ideas from statistical mechanics to help us in this initiative of 4D cellular physiology. So before I keep on going, so you can put my comments in some perspective, Allison told me I should be provocative, so this is my attempt at that. Our group is primarily interested in understanding emergent behaviors in biology using ideas from statistical mechanics. And we've done this, it's not really important what we've done, but we've done this across a large, many different kinds of systems, often in very close collaboration with experimentalists. But we've also spent a lot of time trying to use, understand, and explain machine learning. So for example, I spent most of my sabbatical writing this review you see on the bottom that tried to introduce all these techniques in machine learning in a simple way to physicists. And so what I think I may be in a good position to do is to understand the strengths and weaknesses of both these approaches for trying to perform this kind of 4D cellular physiology project. So at least in my view, what is perhaps the central problem facing us today was summarized by this very nice article by Sidney Brenner when he was giving he was in Nature in 2012, and he was really talking about Alan Turing. But as most of you probably know, Sidney Brenner was one of the pioneers of molecular biology, but he has this quote that I really like, which says, "Technology gives us the tools to analyze organisms at all scales, but we're drowning in a sea of data and thirsting for some theoretical framework with which to understand it. Although many believe that more is better, history tells us that least is best. We need theory and a firm grasp on the nature of the objects we study to predict the rest." And what I think is really interesting about this is that here's an experimental biologist making a case for synthesis. And I think when I look at this quote, what I interpret it as saying is we need a language / conceptual framework for organizing and talking about the measurements of data that are now, everywhere and ubiquitous. And so, when we were meeting to plan this session, we had a big debate about what are examples of such theoretical frameworks. And so I would argue something like population genetics and Kimura's neutral theory and genetics and evolution allow you to make sense of genetic data. Niche theory in ecology is how all ecologists really talk about ecosystems. Attractors in statistical learning theory have played a fundamental role in computational neuroscience, as we saw from the really nice work from Vivek and Anne yesterday. And so the question is, how do we come up with such a conceptual, to me, fundamental question is how do we need a language or a conceptual framework for organizing all these data and measurements? Least is best. So for example, on the left is like, an example for Hydra, which has about 50,000 genes, maybe 20,000 cells. You go measure, you do attack-seq measurements, you measure things in space and time, you can do lineage reconstructions. On the right is this kind of human developmental cell atlas project where people were arguing for something very similar to this 4D cellular physiology. But all these cartoons, I still don't understand what the language is that I'm going to use to understand all these measurements once they're put together. And I think we've seen, that there's at least two kind of very different approaches one can imagine. And one is what I would call an approach based on machine learning and data science. And I think in this day and age, with the success we heard earlier of all these folding algorithms, Rosetta Fold, AlphaFold, with all the successes in machine learning, I don't have to sing the virtues of this kind of approach. We can identify motifs, gene modules, candidate regulators. We can use neural networks to maybe map these different modalities of data. We can learn low dimensional embeddings. This is common, and maybe we can learn embeddings that are, combined measurements. And of course, there's lots of attempts to statistically predict cell lineages and trees for measurements, perhaps integrating location, time, and all these things like that. And so I think in this day and age, I don't have to convince you why this might be a why this is going to be obviously one of the things you should do with all the data and is going to be a central part of any project. But I think it's also worth thinking about what are the drawbacks of this approach? So one of the things we've spent a lot of time thinking about is what can you and can you not do with modern neural networks, and in particular over-parameterized models, which are models where you have many more parameters than data points. And one of the things we've done over this time, and this is summarized in this kind of work where we used a lot of methods from statistical physics, along with Jason, who's going to be talking about a different project, to try to understand how bias and variance work in this high dimensional setting, is that one of the things we are pretty confident of now is that we should think about modern neural networks as something that do interpolation. They're almost like differential lookup tables. And I think the most, and this example is basically, and this kind of idea that they're interpolating and are basically serving as lookup tables means that it's hard for them to generalize to novel contexts, and in particular novel biological settings where you don't already have data. The second fundamental drawback is that we often take this kind of high dimensional data and we use it for visualizing or organizing data in low dimensions. So we often try to visualize it in low dimensions because that's how humans think. And I think Leo Pachter's group has been going around on Twitter over the last month, basically going around and talking about the dangers of basically using these low dimensional visualizations to really make quantitative inferences, like how close are two different cell types to each other, one that you create in vitro and one you create in vivo. And here they came up with an imaging method they call Picasso, and they basically show that rather than doing something like UMAP, you can also like kind of embed them in the shape of an elephant, which is an homage to this kind of Fermi's famous quote about, you give me 3 parameters, I can make a trunk, trunk of an elephant wiggle. So this is one approach, which I think is going to be very powerful, but it does have its drawbacks. And that points to an alternative approach, which I think is grounded much more in statistical physics and that I feel much more comfortable with, which is this kind of idea that what we should do is we should start with biological intuition, because biologists have a language that they use to talk about data, and combine that with ideas from statistical physics and, importantly, experimental data in order to create a new language or framework for thinking about things like cell type or cell fate. And so what I'm going to show you is about one attempt we've done in our group in the last 3 minutes for doing this, which is, most of you have probably seen these famous pictures from Conrad Waddington, who was a developmental biologist in the 1950s, about Waddington's developmental landscapes, where you imagine development using the metaphor of a ball rolling down some surface, and that surface emerging from the interactions of some biological components. And what we've been trying to do for the last decade, I would say, I'm pretty happy with the work, but it's been received only with moderate success, unlike a lot of stuff we've done in other fields, is try to understand if we can now take real data and statistical physics and make this from a cartoon, a drawing, into a quantitative model. And so the basic idea we've actually exploited to do this is to basically use ideas from computational neuroscience / statistical models, like the Hopfield model, where the idea is that we think of cell type as the attractors of some high dimensional dynamical system. And by basically using constructions very similar to those that are used in neuroscience to construct attractors, where you can kind of store memories and the interactions between neurons, we said, let's think about genes interacting with each other, but now instead of memories, you're storing gene expression or ataxic or epigenetic profiles corresponding to different cell types. And using this kind of very simple idea, you could kind of make a cartoon where different cell types, gene expression patterns corresponding to different cells are attractors, and you have a high dimensional dynamical system. In response to signals, you can make these things kind of switch between attractors. And again, it's a cartoon. In this case, it's a mathematical cartoon, but even in the process of making this mathematical cartoon, you learn some interesting things. So the first interesting thing you learn is that it's actually even hard to make this cartoon because we don't have any data on post-translational modifications. So a lot of the signaling that allows you to switch between attractors actually happens through post-translational modifications like phosphorylation, methylation, things like that. And we're largely blind to it. So even though we can put the genetics in, it's hard to put in these things. The second thing you kind of get is that you kind of realize that this kind of way of thinking about it in terms of attractor models allows you to plot data in novel ways that often can correct for batch effects. So for example, on the top, I've shown you some data where we reprogram, we take, reprogramming experiments where you start with fibroblasts, you overexpress a few transcription factors, you turn them back into stem cells. And by using these attractor models, there turned out to be natural, what in physics we call order parameters. And if you plot the order parameters, no matter what kind of reprogramming you do, you can kind of see that all the experiments using lentiviruses, docs, whatever, kind of all follow a canonical trajectory. And this is something you'd expect from general statistical physics. The other thing that I want to point out, I'll skip this middle part for the interest of time, is that it makes extremely novel predictions. So any landscape models will predict that in addition to the attractors that you put into a high dimensional dynamical system, you must have spurious attractors that just occur in the dynamical system, even though you don't want them to exist. And in the context of self-fate models, this is basically the existence of potential self-fates that must exist, that must be supported by the gene regulatory network, but don't correspond to anything you see in vivo biology. And what's interesting is that I have a long time collaboration with some lung stem cell biologists, and I've been telling them this for a decade, and they finally did some experiments, and they saw these kind of long-lasting lung self-fate that doesn't look like any lung subcell type that they've seen before. And they came to me and said, we think we have your spurious attractors, and we're kind of working on trying to figure out how to quantify that, and if that is even true. So I'll just add that there are drawbacks to this stuff. It's much less predictive than machine learning. You really have to think carefully about what data has to be used. As Boris points out, this kind of methods tend to be artisanal rather than scale well. And so I think both these methods kind of are useful. So machine learning is good for many things, interpolation, high dimension, automation, but not everything. And I think what I want to emphasize, the key role for theory is to develop conceptual quantitative frameworks, and more importantly, a language for talking about all this stuff. And at least to physicists like me, it's hard to accept that this will take the form of statistical models. And I think finally, I want to say that once we have this language, we can use theory to guide experiments and think about what gets measured. And a key experimental challenge to me always is, how do we measure post-translational modifications in space and time? Because we don't really have a good way of doing that at scale. And these are the people who funded me and have collaborated and worked on all this stuff. So thank you so much. Stop sharing.
Excellent. Thank you, Pankaj. Provocative. So do we have questions? Maybe while you get organized, I will pitch sort of a softball question. So you mentioned in the sort of single cell analysis, emphasis on visualization. So you project on low dimension because that's what we like to look at. But then talking about attractors, you immediately switch to very high dimensional attractors. So, but you can imagine that there's actually a lower dimensional attractor, or rather there is a manifold, potentially with many point attractors, that actually lives there. So I'm sure you have an opinion on the possibility of extracting low dimensional manifolds from the data via machine learning.
Oh, I think you can. I think you can do it. It's hard. This data tends to be much noisier than you think it is. The second thing I would say is that the way we like to think about low dimension is instead, we have low dimensional things in statistical physics. They're called order parameters. And so what we'd like to plot is basically the order parameters of the Hopfield model. You have different magnetizations. And we found that that actually is much more informative for comparing in vitro and in vivo. It gets rid of batch effects. It does a lot of stuff for you for free. That's what we found. And maybe if the data was less noisy, you could really just go do machine learning. But because we know what we're looking for, we bias ourselves against the noise. And that's one of the things I would say. The bias variance trade-off, I think there's a lot of noise.
Excellent. I see a raised hand. Bin Yu, please.
Yeah, so just a couple of comments. I think dimensionality reduction, like PCA is empirically tested, is still very useful. I just want to make a distinction with Leo. Pankaj was tweeting about was this very flexible dimensionality reduction. There's not theoretical study. It's definitely worrisome, but I think we need to differentiate that with PCA, other things that has been tested. So it's not all dimensionality reduction. It's not useful. Yeah.
I 100% agree. I think it's this over parameterized setting. People are much more confident that, like, look, the stuff you've done with Martin, right? I mean, you've spent so much time like giving us guarantees in many settings, and I would say we have none of those guarantees in the settings that we're using machine learning.
Yeah, I like guarantees because I work, but the problem with guarantees is that it's under also idealized situation which cannot be verified. So again, take a grain of salt, even my own work. But the other thing is the physics, right? So you wrote a primer for physics about machine learning. Can you write a primer for machine learning for statistical physics?
I mean, I would argue that's what Bishop is and David McKay, right? They kind of do that. I don't know. I mean, to me, Bishop is very much a primer about statistical physics too.
So maybe you should put something in the chat box. That'd be great. Yeah.
I think it may take it'll be a very long chat box. So thank you very much. But I think maybe we should zip along. And our next speaker is Anne-Florence. And it's pretty late in the evening for her because she is at EPFL in Lausanne in Switzerland.
So thank you very much, Boris, for the kind introduction, and thank you to the organizers for organizing this great workshop and giving me the opportunity to present. So I'm going to tell you about networks of amino acids and of proteins, and I will talk about two different, All right, so my first part is going to be about collectively correlated amino acids in proteins. And they were introduced in this paper and called sectors. And the idea is that you start from a multiple sequence alignment of proteins that all have the same ancestor, fold into a similar structure, and have a similar function. And you build the covariance matrix of this alignment of proteins. And here you can see in color this covariance matrix actually reweighted by conservation. The red points are the pairs of amino acids which are highly correlated, seems to be quite all over the place. But if you do an eigenvector eigenvalue analysis and reorder the matrix, you will find that there are some groups of collectively correlated amino acids which are along the top modes of the covariance matrix. And it turns out that these sectors found from the statistical analysis of sequence data are constituted of amino acids that are colocalized in 3D, and that have impacts on different aspects of protein function. And so then we may ask where these sectors come from in terms of the function of the protein. And in a recent line of work by the group of Mathieu Viard, it was shown that if an elastic network is evolved in order to be allosteric, that is to say, in order to obtain a certain target deformation on one side as a response to a given stimulus on the other side, or to optimize cooperativity of the deformation, then this kind of sector arises from these materials. And here you can see that the evolved materials also have very particular deformation maps. Like for instance, here you have like one side that slides with respect to the other, so a big shear. And indeed, if you look at the shear intensity, there is a path connecting the two sides with a high shear. So this can be viewed as a material with a soft mode of shear, and other types of these materials evolve hinge or twist design. And this simple model has given rise to some predictions on proteins, such as the scaling of the soft mode stiffness with protein size. And in another work that I participated in, we proposed that sectors can also emerge from selection on a linear trait, so a nonlinear selection on a linear trait. So the trait can be any property of the protein that is selected for, and we make the strong assumption that this trait has linear contributions from all amino acids. But these are the deltas, the mutational effects on a given site. And it was shown that this nonlinear selection on additive traits describes well experimental data in this paper. And in this view, we can define a sector as the set of sites which have the large values of mutational effects on the trait under selection. So this trait is very abstract, but a concrete example of a trait is the elastic deformation of a protein that changes between two important states. If it's written within a small deformation and small mutation effects, then this deformation energy can be written as a linear trait. So that's just a toy example. And then we can perform selection on these linear traits, and we perform nonlinear selection, for instance, with this quadratic function. So here we have a histogram of the values of the traits for all types of random synthetic sequences. And we put a selection window around a preferred value of the trait that we assume is preferred by selection. And then we can analyze the resulting sequences that have gone through this selection. And we started by doing an eigen decomposition of the covariance matrix inspired by the first study on sectors. And we found that there is a strong outlier in the spectrum of this covariance matrix, but it's rather the smallest eigenvalue than the largest. And we found out that the associated eigenvector actually reproduces extremely well the vector of mutational effects delta. And so it can give us the information about the sector, which is the set of sites with the large delta. If we go back to the model that we used, in fact, we can understand where this comes from. It's because the selected sequences satisfy this constraint that the scalar product is close to the ideal value of the trait. So if we represent them in an abstract space, the selected sequences have to lie around the plane, which is perpendicular to the direction of the vector delta. So it means that delta is a direction of particularly low variance, depending why it's associated to this lowest eigenvalue. And in fact, because, for instance, Pankaj told us about Hopfield models, this is in fact deeply related to a Hopfield model. Delta is a repulsive pattern in a generalized Hopfield model. Okay, so we also introduced a more robust method in order to find the sectors, in order to address the fact that there may be other directions of low variance in the data. And I won't go into the details because it's a bit technical, but just to summarize a bit. So far, we've discussed the sectors of collectively correlated amino acids in sequence data. We've shown that they can arise from soft modes and also from nonlinear selection on a linear trait. And I would like to mention that it has also been shown recently in this paper here, that in the presence of a soft mode, each mutation deforms the protein along that mode, and this results into nonlinear selection on a linear trait. So these things are really related. And yet another paper showed that a very related result, noise and mutation-induced deformations of the proteins are restricted on a common low-dimensional subspace, which gives rise to this kind of global epistasis. So to summarize on this part, I would say that in this small area, theory can be really viewed as an organizing principle, which showed us the importance of collective soft modes and nonlinear selection and additive traits on the sequences that we can observe, the data that we can observe. And now I would like to say a few words also on how we can infer which proteins interact together from sequences. And so this is a network of proteins. So at another scale, not a network of amino acids, but a network of interacting proteins. And it's very important to understand it because protein-protein interactions are crucial in cells, but their experimental determination is still pretty hard. And there are so many of them that we would like to find out that it's a tedious thing. So it's interesting to find ways to determine them from sequence data. And many methods have been developed for this. This is just a set of examples, not exhaustive at all. I just want to emphasize that there are different types of methods. Some of them are based on phylogeny, others are based on sequence coevolution, and yet others assemble all types of evidence, including literature curation, experiments, etc. And yet others are machine learning and classifier based, including deep learning. Okay, so that's just a very fast overview. I would like to say a bit more on one of these methods that I've been involved in the development of. And so the basic idea of this is to use the fact that if two proteins interact together, then their amino acid sequences are going to show some correlations. And it's due to the fact that amino acids in contact need to be complementary in terms of physics and chemistry. If a mutation arises on one side that disrupts this complementarity, then it's going to be selected out by natural selection, and it can be restored by another mutation. That's a very simple sketch, but it gives the idea of why there are these correlations, and it was also mentioned earlier by Min Beck in her talk. And so here, we are going to use this in order to find out which protein A interacts with which protein B in a given species, where A and B are two given protein families. This question arises because there are often multiple paralogs of a given protein in a given genome, so multiple copies from the same family that are similar but not the same. And we want to employ patterns of correlation. So for this, we started from a model which was developed in order to predict structures from sequences, which is called direct coupling analysis in the field, but is also, in fact, more generally a maximum entropy model, pairwise maximum entropy. So the idea is that if you have a training set of known interaction pairs, you can measure the one- and two-body frequencies of occurrence of amino acids and construct the associated pairwise maximum entropy model. And it turns out that these couplings here are much better predictor of 3D contacts than just the covariances, Cij. And we employ these couplings in order to build a score for each possible pair in each species. And the score is just the sum of the pairwise couplings. And we developed an iterative algorithm which approximately minimizes these effective interaction energies between partners. And we showed that on a benchmark data set of proteins, it's able to make good predictions of protein-protein interactions. But something that we also noticed is that these correlations can come from what we initially thought of, which are the correlations from optimization to preserve the structure and the contacts. But they can also come just from phylogeny. Even if all mutations were neutral, so Pinus landscape completely flat, if you had mutations that, for instance, occurred early in the history of the protein family, then like these two mutations here, you see like EC, this would give rise to correlations in the sequence data. And so we asked in a synthetic data set, what happens if we have only neutral mutations and only phylogeny? And it turns out that this score, which is based on the maximum entropy model, is able to predict which proteins have co-evolved on the same branch, even in the absence of any selection for structure or function. So this shows the importance of phylogeny, and in my opinion, it also shows that the methods which are disjoint, some on phylogeny and some on coevolution, should be taken together. And I will also just point out that phylogeny is also important for sectors, even though I don't have the time to say much more about this. And with this, I will just conclude that for this question of inferring interaction partners from protein sequences, we can ask, in fact, different sub-questions. One of them is just to make predictions, and for this, probably methods based on deep learning are very useful, but we can also ask more fundamental questions, such as what is encoded in protein sequences, what are the signatures of interactions coming from optimization or historical contingency, and for these questions, principled methods based on physics are more appropriate, I believe. So with this, I would like to thank you all for your attention, and I also include this acknowledgement to all my colleagues who worked with this worked with me on this project, and also my current group members and my current funding sources. And I'm happy to take questions.
Wonderful talk, Anne-Florence. Thank you.
Thank you very much.
Let's see if we have any hands up. Not quite yet. So you talked briefly, you mentioned soft modes. And I'm just so wondering, so there are two kinds of soft modes, right? So one can think of soft modes in, if you like, genetic in sequence space, which are sort of patterns of variation or covariation. But there are also soft modes as sort of mechanical soft modes.
Yes, that's a good point.
And I had a feeling that you wanted to tell us something about it, but you didn't. Could you?
Okay, so like the part I mentioned on the allosteric material is really meant about the mechanical soft mode. So if you select for optimized allostery, it gives you these mechanical soft modes. But then I also mentioned that in fact there is a relationship between like the space of deformations of the mechanics of one given protein and the space of deformations induced by mutation. If you make the assumption that the mutations have small effects, then it can be shown that there is a mapping between them. So that's the paper by Arvin Yurigan, for instance, that showed this, and also the one by Kuniko Kaneko. Yes. So there is a relationship between them at least under certain assumptions. And the covariance matrices are quite similar, in fact.
Thank you. Very interesting. You went fast, but worth reading about. Armita, you have your hand up.
Yeah, so let me lower my hand first. Okay. Very interesting talk. So there are all these connections between covariation in the proteins that we observe out in the world and sort of models of proteins that you evolve synthetically and try to look at, physical soft modes, impact of mutations on that, how that translates to epistasis, Arvind's work or others. So here's a question: is sort of having mutations tuned to mechanical soft mode somehow an indication of protein? So the ensemble of proteins that we are observing are rather evolvable, right? Because we are looking at some biased ensemble of survived proteins that have survived through evolution.
Oh,
right?
yeah, indeed.
And right, and so the sort of the fact that something is evolvable it needs to be somewhat robust to mutations, but also be able to adapt to maybe new conditions and so on and so forth, right? So that introduces some constraints in what type of mutations you can have, and maybe modularity or the sectors we have, maybe one of the features that would emerge just by asking for evolvability. So can you sort of, I'm sure you have thought about it, can you sort of expand on this? Because that to me, it seems like a deep theoretical question, as like, what's the structure of protein universe?
Indeed, I think it's a super interesting question. So the point about modularity, I believe that there is a study by Olivier Ivoire who also showed that this modularity can evolve depending on the environment variability during evolution. So indeed, it's deeply related to this evolution and evolvability question. And I think the soft modes are also something that is deeply related to this evolvability, because a mechanical soft mode is something that doesn't cost much energy, and as such, it's a very interesting thing from the evolutionary point of view. And if it and the fact that it's very collective also means that it's, it can be in principle modified, but at the same time, studies have shown that the soft modes are quite well preserved in protein families, showing that probably there are some constraints to maintain them because they are so useful. So yeah, I mean, I think they are both, both rather robust to mutations, and yet can like, for instance, yes, while the soft mode is quite robust, some you can imagine that some pieces of the protein could nevertheless move more. So I think the collective thing is rather robust, but some local ones there. Yeah, but I think it's a super interesting theoretical question.
I think in the interest of time, we should move along, Armita, and I think maybe it's your turn to take over the control here.
Thank you very much.
I'll start. Thank you, Anne-Florence. So our next speaker is Isabella Graf from Yale. So Isabella, if you're ready, take it away.
Cool. Can you see the slides?
Yep.
Okay, awesome. Cool. Thanks a lot for the introduction, and thanks, of course, also to the organizers for giving me the possibility to present our work here at this, I guess, really stimulating conference. And today I would like to talk about our recent results on the phase behavior of multi-component mixtures with structured interactions. And in particular, I guess I would like to show you how I think theory provides a really nice way to systematically reduce the complexity of these high-dimensional systems to a much lower dimensional representation. And I guess that also this somehow offers a new perspective, or also a new framework to think about these types of systems. And so the phase behavior of multi-component mixtures is, I think, very relevant in different contexts of biology. And as you've probably all heard, liquid-liquid phase separation has been suggested as a type of organizing principle for cells. the idea is basically that via phase separation, cells can actually compartmentalize their cytosol into so-called membraneless organelles or liquid-like droplets without the need for really like boundaries like membranes. And then in a somewhat similar spirit, I think it has been shown that the lipid composition of cell-derived plasma membranes is tuned close to a special point in phase space, a so-called miscibility critical point. And at this point, you have very long-range correlations and very high fluctuations. And it's kind of, in both these systems, you really have many different types of components, like many proteins and DNA, or many lipids and proteins that somehow interact. And somehow the phase behavior of this complex system seems to play a very important physiological role. And I guess at the same time, what is maybe surprising is that it's quite unclear what the right control or order parameters are in these systems and how cells could actually navigate in these very high dimensional spaces. And so I guess the question that is kind of underlying my talk is, what is generally the phase and critical behavior of multi-component mixtures? And typically, if modeling these multi-component mixtures is done in what is called Flory-Huggins theory, and basically this is a mean field description of these mixtures, which takes into account two different contributions. One contribution from the entropy of mixing, and then an energetic contribution, which is due to pairwise interactions between components, which are characterized by this interaction matrix chi-ij. And I would say there are like two limits, which are quite well understood. The first one is if you have mixtures with only few components, like let's say binary or ternary mixtures. And in these cases, this free energy is embedded in a quite low dimensional space, either 1 or 2 dimensional. And it is really possible to predict the phase equilibria in this landscape by what is called a common tangent construction. But now, if you think back about what I told you about the biological systems, they are typically not composed of 2 or 3 components, but rather of really many, many different types of components. And in these cases, these common tangent constructions, they become really infeasible because then the free energy would be embedded in a very high dimensional space. And so one approach that people have taken to still model mixtures with many components is by approximating the interactions between the pairwise interactions between components as random numbers, which are drawn from a certain distribution with a certain mean and variance. And in this case, it was shown that then the phase behavior indeed only depends on this mean and variance of the interaction strength. But now again, I guess we would expect that really the interactions in biological mixtures are not really random, but they are basically due to physical characteristics of the components, like for instance, electrostatic interactions or let's say some hydrophobic effects. And so I guess we want to, in this project, take a kind of complementary approach where instead of looking at mixtures with random interactions, we want to understand how the structure of interactions shapes the phase behavior. And so basically the question that we are trying to address is if, depending on the structure of interactions, we can somehow systematically reduce multi-component mixtures to a lower dimensional representation. And to do so, we look at a multi-component model with, in principle, an arbitrary number of components n. And the idea is that each component is characterized by R different features. So in the case of lipids, this could be like the lipid tail length, maybe the head group or the saturation. And so we have these components that are characterized in this case by four different features. And then the idea is that each feature somehow conveys an additive Ising-like interaction between neighbors. And I guess what is nice about this model is that it, in a sense, allows us to really systematically investigate the role of the interaction structure for phase behavior. And what I mean by this is that if you look at the Flory-Huggins theory for this model, what we find is that the pairwise interaction matrix is non-random in a very specific way. And it is non-random in the sense that the rank of this matrix is always less or equal than the number of features. And so basically by changing the number of features, you can somehow change the structure of the interactions. And what we do is then we derive analytic conditions that tell us if the phases are stable against local perturbations, or also conditions for critical behavior. And what we find is that these only depend on the correlation structure between the features. So basically how distributed the features are among components and how varied or how correlated they are. And to be more quantitative, what we find is that what matters is this, what we call feature covariance matrix, which is really the covariance between features gamma and beta. And then all averages are taken with respect to the mixture composition. So basically with how often each component occurs in the mixture. And what this tells us is the following. So here is kind of an illustration of a mixture with 500 components and 2 different features. So each dot corresponds to one component. And in this case, this is a kind of randomly generated mixture, but it's not a random interaction matrix, but instead the mixture is generated from a multivariate Gaussian with a certain fixed covariance matrix. And what the idea is basically is that instead of looking at a 500 dimensional space of all the densities, it's actually enough to look at the system in this 2-dimensional feature space. And that basically what matters is only the distribution of components in this 2-dimensional space. And to be a bit more specific, what we find is that if you want to figure out if a system is thermodynamically stable or not, then what only matters is how the distribution is of these components in this low-dimensional feature space along the first principal component of the covariance matrix. So basically we have these 500 features, they have a certain covariance matrix, and then to determine if the mixture is stable or not, we look at the first principal component, which is shown here in gray, and then if the variance exceeds a value of 1, the system would phase separate, and if it's below 1, it's stable against local perturbations. And in a kind of similar spirit, we can also derive that if we look at the initial direction of phase separation, or basically how enriched a certain component is right at the spinodal, then this is just given by the feature vector of the certain component projected onto this first principal component. So basically, this is what is shown here as a color code, that the relative enrichment or depletion of the different components only depends on the location of these components projected onto the first principal component. And in terms of this relative enrichment, we can actually reformulate this criterion for thermodynamic stability by saying that the system is marginally stable if the variance of this distribution of relative enrichments is exactly equal to 1. And similarly, what we find is that if we want to consider critical behavior, then as an additional condition to having that the variance needs to be equal to 1, what we find is that the third moment of this distribution of the relative enrichment should be equal to 0. So this is basically saying that the skewness of this distribution has to be equal to 0. And I think this kind of nicely generalizes results for binary mixtures, where it has been found that critical behavior occurs right when the mixture is completely symmetric. So basically if the 2 phases are symmetric. And I guess this makes it more specific in a way that it says that this distribution doesn't necessarily have to be completely symmetric, but it has to have a 0 skewness. And overall, I would say what is nice is that maybe our results suggest that really what matters is this distribution of components along the dominant eigen direction of the feature covariance matrix, and that this is really what determines thermodynamic stability and critical behavior. And with that, I would like to sum up. So I think I hope I could show you why we chose this certain multi-component model to investigate how the structure of interactions really shapes local thermodynamic properties. And what we find is that really stability of phases and critical behavior depends just on the distribution of feature or components in this feature space. And since we would typically assume that the dimension of this feature space is much lower dimensional than the number of components, I think this really allows us to reduce the complexity of these systems to a lower dimensional matrix. And maybe in terms of experiments, since I guess this was one of the questions, I guess I've talked a lot about these distribution of these relative enrichments. And I guess these are kind of like partition coefficients you could measure in experiments. And so maybe I think one set of experiments which would be nice is to really measure different partition coefficients for different mixtures. And maybe this would give a way to say more about the interaction structure or pairwise interactions. And with that, I would like to thank Ben, with whom I did this work, and the whole group, and of course, you for your attention. And I'm open for questions.
Yeah, thank you, Isabella, for this inspiring talk. So I can see Pankaj has his hands up. Maybe just unmute yourself and ask the question.
Yeah, I was just wondering, this was really great. It was really fun. I was wondering if you actually just took your low rank matrix and added noise. You know, from like just statistics, we have these, you know, tensor plus spike plus tensor models, and we know that if you add a little bit of noise, you basically look random. So I was wondering if you could, if a similar intuition would happen, you know, would apply here, that if I add just a little bit of noise, enough noise per component, that actually it'll look much more like random than it would look low rank. Or do you have any intuition about that? Because usually there's a phase transition between those two with just tiny bits of noise, unfortunately.
Right. So yeah, that's a good question. Actually, I think there has been a paper put on arXiv like only a few days ago about this point, that what happens if you have like random interactions and deterministic interactions. And in this case, I think what they found is that it depends strongly on basically the eigenvalue spectrum of the deterministic part and the random part. And that basically it's about if kind of the additional randomness makes it that the eigenvalue is kind of below the one of the deterministic part or somewhere above or in it. And depending on this distinction, either the randomness doesn't really matter so much or it has a very strong effect. And so I think this, I'm not sure if I can really say very generally what the effect would be, but I guess it depends on kind of how the eigenvalue spectrum changes.
Okay, great. So we have a question from Q&A. Oleg Igoshin is asking, well, they're saying nice theoretical insight on formalism, but is there a particular puzzle in the existing data that you can explain with this?
Oh, okay. So I guess, so I would say, I don't think that we came up with this model to really describe a very specific system. But I think what this theory provides is kind of a new way to think about about generally like multi-component mixtures where you have this problem that you don't know what like right control parameters are or like order parameters. And so I think in a sense it provides a kind of guidance on what makes sense to look at and how you could design experiments to figure out more about interactions and things like this. In terms of, I guess maybe one point is that if you have these random mixtures, then I think there's typically what people do is that they look at mixtures with an equimolar composition. So basically all the components occur equally often. And I think in this case, if you look at critical densities, then these densities diverge in the limit where you have many components. And this is something that you don't have here because we looked at also cases where not all the components occur in equal amounts. So this is, I guess, something that maybe previously wasn't so clear, but this is also something, for instance, in this new paper that they have where they combine deterministic and random interactions, what they also find there that just by having like some kind of deterministic interaction part, you can actually avoid this problem of this diverging critical density.
Okay, I think in the interest of time, we should move on to the last talk of the session. And our last speaker is Jason Rocks from Boston University. Jason, if you can share your screen.
Did I get the right screen or did I get the wrong screen?
Wrong one.
The wrong one? All right.
Yeah.
Can never predict which one it will give me. Here. Did that work this time? No?
Yeah.
Yes.
Yes?
Perfect.
Okay, great. Cool.
Yeah. Take it away.
All right. So, yeah, so I'm a postdoc with Pankaj at BU. So I'm going to be telling you about thermodynamic models of post-translational modification. And as kind of a side goal, what I would like you to take away from this talk is that theoretical models can also be useful at a very practical level. For example, by telling you how to even look at complex biological data, like how to actually represent the data in a way that you can easily identify where the biological signal is as opposed to just experimental noise. And so, so one of the big open problems in biology is understanding how eukaryotic cells can sense and respond to their environments. And so one of the main ways they do this is by using protein signaling pathways, which are triggered by external stimuli, which then create a cascade of interactions that communicate information to the cell about the environment. And then once the cell has this information, it can use it to regulate important processes like cell division, growth, metabolism, and things like that. So, excuse me? All right, I guess it wasn't anything. So these protein signaling pathways are composed of complex networks of protein-to-protein interactions. And many of the interactions between proteins take the form of post-translational modifications like phosphorylation, methylation. So historically, the way people have gone about studying these pathways is by trying to identify all the components involved. So identifying all the different proteins and trying to quantify all the interactions between them. But our colleagues at the Bushore lab at Rice decided to take an alternative strategy where instead of trying to map out all the pieces of existing pathways, they wanted to see if they could understand such pathways by building their own, and more specifically by trying to build some of the, create synthetic versions of some of the molecular circuits that are involved in these pathways. So for example, so for example, so this is a kind of a standard push-pull mechanism where there's some substrate which can be phosphorylated by a kinase enzyme or dephosphorylated by a phosphatase enzyme. So here's what these are. Oh, good. All right. And so the way that they create these circuits is by transfecting embryonic stem cells with DNA to express all the different components. And then once a cell is successfully expressing all the different components, they then introduce fluorescent antibodies that can measure the concentrations of all the different pieces. So for example, in this case, they can measure the concentration of kinase, phosphatase, the substrate, and then also separately measure the concentration of phosphorylated substrate. And in these systems, they can control various properties like the concentrations of all the different proteins, the binding affinities of the different enzymes with the substrate, and also the phosphorylation or dephosphorylation rates of the different enzymes. And then their goal is to eventually construct a modular toolkit to design complex circuits with very specific behaviors. So here's an example where you can imagine daisy chaining some of these molecular circuits together to create a much more circuit with much more complicated behaviors. So for example, you have a membrane receptor, which when activated, activates a kinase enzyme, which then phosphorylates a substrate, but then you can imagine that substrate also being a separate kinase enzyme, which then phosphorylates a second substrate, and so on. However, if they want to design such circuits from individual pieces with very specific behaviors, then they need a way to actually predict what's going to happen when you put these different components together. And so this is where modeling comes in. And so two things we have been helping them accomplish is to infer the different biophysical parameters from experimental data, like binding affinities and phosphorylation rates, and then predict how these circuits will behave once we, in new contexts where we combine the different pieces together. So now I'm going to walk you through a very simple example where we've been doing this with some success. So here's a really simple push circuit where you have a kinase enzyme which can phosphorylate a substrate, and we can. And so what they do is they express this construct in a large number of cells, and then for each cell they use these fluorescent antibodies to measure the concentrations of the kinase, the substrate, and the amount of phosphorylated substrate. And so here's what the raw data looks like. So on the x-axis, I have the kinase concentration. On the y-axis, I have the substrate concentration. And then what I've done is I've created a histogram and colored each pixel in the histogram according to the average concentration of phosphorylated substrate. And so if you look at this data, it's actually not obvious that this construct is even working, because because you don't even know what it's supposed to look like. So for example, it could just be that, so you can see that as I increase the amount of kinase, the amount of phosphorylated substrate increases. But it's hard to separate that from the just total amount of substrate. It could just be that as I increase the amount of substrate, I also get more phosphorylated substrate, because ultimately you need to have phosphorylated substrate, you need substrate. And so to analyze this data, we've been creating these kind of quasi-thermodynamic models. And so one complication you have in modeling these molecular circuits is that while some of the interactions you can think about as being equilibrium interactions, where you would normally be able to write down a partition function, other interactions like phosphorylation are inherently non-equilibrium interactions. But the way, so you can't write down generally a partition, like a standard partition function for these systems. But it turns out what you can do is break these systems into pieces which individually behave like nice equilibrium systems where you can write down partition functions. So for example, I can write down a partition function for how the kinase binds to the unphosphorylated substrate. Separately, I can also write down a partition function for how the kinase binds to the phosphorylated substrate. And then what I can do is link these systems together using the non-equilibrium interactions by assuming that they're steady state and then writing down a steady state equation. And so what this then allows you to do is combine the two models for these equilibrium systems into kind of one big model and it turns out what you get is kind of something that looks like a partition function. We have the weighted probabilities for the two different pieces, but it's actually not quite one. But a useful thing that comes from making these models is that you immediately get a prediction, and that's that the fraction of phosphorylated substrate should actually only depend on the ratio of the kinase to the substrate. And so to see if this is what the experimental data is doing, I can take a bunch of horizontal slices through this data and plot each of these slices. So here now the x-axis is the amount of kinase, and then the y-axis is the amount of phosphorylated substrate, and then I've colored each curve according to the total amount of substrate. And then if I just rescale the axes, you can see that all the curves collapse. And this is not something that's obvious by looking at the raw data. So you can see now that the fraction of phosphorylated substrate clearly depends on the ratio of the kinase to the substrate. And so what you can see here is that the theory has, like, even before we've done any inference about biophysical parameters, it's already facilitated the way we go about analyzing data and even determining whether the experiments are working. And I also just want to let you know that it's actually not obvious that this data should just magically collapse, because there are some other basic problems which we really had to work on here, which are, what are even the units for this data? Can you even, if these are, you have antibody measurements for the different components, like how do you know that they're all representing concentration in the same way. So there's a whole bunch of extra control experiments we've had to do. But it's interesting that the theory still makes, pretty quantitative predictions, even though we haven't done any post-processing of this data at this point. All right, and so I just want to, this is a lot of speculation, but one of the next steps you could imagine doing is using deep learning models to kind of hone in on specific parts of these experiments. So for example, if you wanted to make predictions about what specific sequences of, say, kinase will result in specific binding affinities between, say, the kinase and the substrate. And so this is a place where deep learning models could really make a big difference, where then you use the model, deep learning models to predict different biophysical parameters from sequence data, but then you combine them with, you then use these biophysical quantities to make predictions with biophysical models. And so you can really see how you can combine these learning models with biophysical models. All right, so thanks for your time.
Yeah, thank you, Jason. Is there any questions out there? Just raise your hand. Just to start, I'll have some. I have a question. Maybe you mentioned and I missed it. So the exact structure of your, what you mentioned, a circuit model, right? So the two compartments with non-equilibrium interactions between them, right? So how did you decide that that's the structure of the circuit model you want to use? How many iterations you have gone through to land on that one?
Well, I mean, basically we just started with the most general model where you know that the kinase binds to the substrate, so that's one interaction. And you know that when the kinase is bound to the substrate, it phosphorylates it. So that's kind of the second interaction, and that's basically all that goes into it. And once you know that, you just, there's actually a very systematic way to start breaking all the different states, so all the different combinations of ways things can bind together into different parts that look like, that behave as if they're in equilibrium. And then you simply, just draw an extra connection whenever you see a reaction that's not in equilibrium. And you infer that interact. So basically, yeah, I mean, that's all I can say. You just look at every possible interaction. And since in these experiments, they're designed to have a very limited number of interactions, that's pretty straightforward. And then, I mean, that's basically it. So.
So can you in principle quantify how much this system is driven out of equilibrium? And
I mean, kind of by definition, it's not in equilibrium, but we are assuming it's in steady state.
Right.
So, right, in principle, kind of, if you wanted to, you could also write down, all the kinetic equations for all the interactions that could happen, and then, set all the kinetic equations equal to 0, and solve those equations, and that would be the kind of an equivalent thing. But it's a little bit more interpretable if you separate the network, the system, into these little pieces that you can talk about as being equilibrium. You can write down partition functions, you can write down the probabilities of the different components, and then you stitch them together with the interactions that they've had to leave out.
Okay, so we have 2 questions in Q&A. Ibrahim Olsenman is asking, well, great talk. Is this general for any kinase substrate system? So the model you're proposing.
Yes, I mean, pretty much. I mean, this is such a, I mean, I don't want to say simple, but there's only one interaction or 2 interactions in this kind of push model. Now, of course, if you have, as soon as you start introducing other components that combine to them, then it gets much more complicated. So, but I mean, we made a lot of strong assumptions here, right? That the kinase binds to the substrate in a way that's in equilibrium, but then the phosphorylation takes place in steady state. So,
And the other question by Oleg question is that, is there some implicit timescale separation behind this representation that you're talking about?
Yes, I mean, we're assuming that all the interactions happen, very quickly. That's basically it. That whatever the timescales are for the cell to actually express all the different components, we assume that is, the rate of change that the concentrations might actually change. We assume that that's much slower than the actual interactions between components.
Okay, great. I think we can basically move into our discussion session of this last session. So I'm not sure how to do that. Janine, maybe you can help us with this.
Yep, we are all set. We're going to bring everyone back on.
Okay, perfect. Boris, do you want to start, or how should we do this?
I suppose I could. Well, so it seems to me, so our session ostensibly was on networks, and right, everybody talked about the networks of interactions, but to me, it really what came through was this theme of dimensional reduction, right? And now I guess I already asked Pankaj about that. So one aspect of it, right? So in this single cell data, there are all these algorithms that kind of aid visualization by finding some suitable variables to project. But of course, everything, if projected on 2D, will become two-dimensional. But one can ask so a deeper question: what is sort of the intrinsic dimension of the data? What is, could there be actually a hidden by noise, but low dimensional manifold? For example, in the context of these Weddington attractors and cell types, I can imagine that this attractor might, these clusters, point attractors might still lie on low dimensional manifold, let's say of 4 famous developmental control factors, some uber regulators that slave all other degrees of freedom. And then there is some, it acts as a center manifold. There's some sort of dynamics that unfolds within that manifold. And one question is, can we just discover it by, data mining, machine learning? But then on the other hand, right, there are all these ideas coming from physics, so collective modes, soft modes, and in Anne-Florence talk, and also in Isabella's talk, there was also dimensional reduction, right? So for soft modes define kind of relevant variables that ultimately, at least locally, will show up as principal components. But curiously, there are also seemingly sort of more relevant modes in the parameter space. In Isabella's talk, right, these were parameters in the model. So, the dimensionality in the parameter space was reduced. In Anne-Florence talk, it was dimensionality. So, there were these correlated modes of genetic variation. But can we develop this if this is not orthogonal to what you think? Maybe we can discuss it a little bit. Or if it is, then we can discuss that too. Pankaj.
I'll just say, I think this relates to something that Hernan and other people were saying in the last session, which is that perturbation is a very, very powerful tool in biology. And we know in physics also, the way you discover collective modes is you do perturbations. The stuff that decays the slowest to perturbations is our collective mode. And I think in a place like Genelia, that seems like a dream, right? But at a place like Genelia, you can imagine making big perturbation response matrices. And then measuring these matrices, the kind of matrices we're interested in, me and Jason, Anne-Flo, everyone, Isabella, and using that to actually find the collective modes. Whether they'd be hidden by noise, I'm not sure, but I think perturbation is the key to identifying collective modes and measuring what we in physics would call susceptibility matrices, right? And so again, this hints at the kind of thing Genelia is really good about. Like if we can make really controlled perturbations, then we should be able to get at these collective modes. And the theory would give us a language to talk about these collective modes, right? That's supposedly the collective modes are the same, and then we have a phase transition, we go to a different kind of, qualitatively different kind of collective mode. And that would be the hope for me. That is what I would say is how we would understand what really, to me, a successful 4D CP program would be. Okay, we know the collective modes of this and that, and how they're coupled to each other. I guess that would be my very high level answer. Perturbation is the key. And then not being blind to stuff too. Like I really, really feel like we're blind to post-translational modification. We pretend it doesn't exist when it is, I would say, at least half of development.
So I see raised
yeah. So just for clarification, it seems like there are people who know both the physics perturbation and biological perturbation. Does the physics perturbation inform the experiments to do biological perturbation? Is that at that level we're talking about? I'm just trying to understand. It would be nice, like they use the same word, there might be some high level correspondence, but are we at the level of the mathematical perturbation in statistical physics actually inform some biological, like what interventions you would do?
Pankaj, you want to take that?
I mean, we have certainly tried. I mean, I can just tell you, we ran into a sociological problem. The last time we tried to do this, we're trying again with our stem cell. Yes, I think it depends how much you believe the theory. We certainly believe them enough to convince some people to do some expensive experiments. That's not, like whether they work, I can't tell you, but I consider that a success of theory.
Do you know the result? You're waiting for the results, or they haven't panned out yet?
No, we're starting the project again. I kind of abandoned the stem cell field for 6 years and came back to it recently because my experimentalists came to me with one of the things I said you should see, and they were excited. So I abandoned it for sociological reasons. I found that I don't want to get into, but I found it was hard to do theory in stem cells and much easier to get credit to go back to another theme throughout. If I ran the same kind of arguments in microbial ecology, I got a science paper. And if I did it in stem cells, everyone called me a crank and I had to call in favors to even get papers published. Thanks, Oleg.
Well, sociology, huh? Michael.
Yeah, so I just wanted to jump in on the perturbation idea, just because actually historically we did a lot of this at Genelia. You know, it was an interesting idea to do neuroscience a different way, right? We had some new tools. I would say the normative theories of like what should happen when you start breaking neurons were pretty straightforward. So we did a lot of it, and I would say it was expensive and time consuming, and it was interesting, right? But I think that is actually the challenge and really the maybe testing Pankaj's idea of like whether the theory is successful, right? Is that basically you're going to ask a lot of people, tens of experimentalists to line up, right, to test an idea, right? And so it had better be a good idea, and there had better be some kind of bootstrapping, right? Where initially you say, okay, here are some really good test cases, right? And here's a really well worked out theory, and you kind of have to map out all the ways things can go wrong, right? And it's going to take 5 years, so there'll be plenty of time to think and reevaluate these ideas, right? But I think in general, it's a great idea, right? Like to really lay out a whole vision, which sits on a bunch of perturbation-based ideas and measurements. And initially, the measurements aren't going to be good enough, but over time, things will get better. Hopefully, the perturbations become more precise as the questions get sharper, right? So I think it can absolutely happen, right? But it's going to be like a really big gamble, right? And so we need a pretty confident starting point. Right. So if anyone has a really specific idea or vision for it, because conceptually, I think many people agree it's wonderful. We should do it. it's going to be expensive and kind of painful. So several people need the courage to line up, I think, behind that vision.
So
Okay, but
how-
we're kicking the ball back into experimental court, right? And this is a theory workshop. So I'm a little surprised that I don't seem to get any takers from machine learners and data miners to comment on the feasibility of learning manifolds with a certain degree of confidence from the data.
I think we have Genevieve as a data miner and machine learning person. Yeah, go ahead.
Actually, it's really funny because I was thinking that about 30 seconds before you called us out. But yeah, we have a ton of data. And maybe I didn't make this super obvious when I said like cells are the basic biological unit of life. So the inherent perturbation exists in that there are lots of different cell types. So if we can borrow information across what's been studied in the lots of different cell types, and I mean, if you want to, sorry, you're talking about collective modes. I mean, just look in the human fetal cell atlas if you want to do it in gene expression data. I mean, you could potentially, I mean, it gets back to David's labeling problem, and there are issues. There's lots of caveats, and I'm not saying it's going to be easy, but there's also lots of data that already exists that can be mined to make preliminary evidence for whatever theory you're hoping to test. And I will say, one of the things that really drew me to transfer learning and has, you know, I'm just continually amazed again and again, is how generality, like the ability to generalize, the ability to take something that you would never expect to, you know, you learn in one system and then it's in another system too, that's totally unrelated or totally different, you know, molecular species, but it just comes down to, you know, tuning the temporal dynamics between the two. Like it A, teaches you something, and B, like it really helps guide you towards what should be the next experiment and what should be the next question. So I think before you, like, there's definitely huge opportunities there.
So one thing I wanted to add, I mean, in some ways it connects, in some ways also opens another sort of area that we have been talking about throughout this workshop. So pointing at one of the questions that we talked about today. So Anne-Flo talked about proteins from sort of physics-based approaches. And before that, we heard a Rosetta Fold and AlphaFold talk from Minkyung, right? And so this is approaching the question of proteins from two different directions and trying to also, you know, an AlphaFold has used a lot of ideas that were previously developed by theorists. Now, the question that I have is, well, there's a lot done on the computer science and sort of machine learning approaches. So, but what's the interplay between the theory and machine learning in a sense to learn biological systems? How we get at designing better perturbation experiments to go and after sort of functional responses in proteins and try to sort of learn, you know, structure is not the end, right? To learn basically a map to function of a protein, to map out, you know, ideas about evolvability of proteins and general protein space, right? Yeah, so basically the sort of conversation between theorist, machine learner, and then eventually experimentalist, someone convincing them to actually do something and try to test these hypotheses, right? Is there a sort of path that we can see? Because as far as I can see, machine learning is here to stay and it's a very powerful tool. So we better better integrate our theoretical understanding with these sort of approaches to make sense of it. You know, and Flo, if you have some ideas about this.
Yeah, so thanks, Armita, for raising this very interesting on topic, on the like current topic. So yeah, I mean, I would say it's completely true that methods like AlphaFold have built on the understanding that was gained by, for instance, co-evolution methods. However, I should probably say that the last, like AlphaFold 2, has actually resorted to using directly full multiple sequence alignments rather than pairwise features such as decouplings, EIJs, right? So I mean, it means that like it has been definitely useful, and it is still useful in some of the models, but you know, in AlphaFold 2, it's not the crucial point. So you know, there are some, yeah, it's a question that is still, I think, very open. I would say, to me, the key point is that machine learning and deep learning methods, especially deep learning ones, have to address some question that is super well posed with an objective function. And there are other types of questions that we can ask that either are not yet to this stage or are not to this stage. And I don't really know which one is you know, the real case, but at least not yet. And for instance, I would say that for me in the field of proteins, I mean, although there are these giant data sets which are really amazing to look at, etc., something that has really taught me a lot in practice is constructing some synthetic data sets with very simple rules and seeing if these kind of behaviors are found in real data. So I would say that, again, it really depends on the question. If your question is to predict structure, I mean, okay, I mean, it's already clear that deep learning is a great method to do this. If your question is more understanding how function is encoded in a sequence, then it's a question that, as of now, is more abstract and doesn't really have an objective function. So yes, I would say it's rather the type of question, but I think these deep learning methods are also amazing and make great predictions.
But
for,
maybe one
yeah.
can so sharpen this question about the functionality and pardon my ignorance. So machine learning tools can predict the fold. Can they predict, if you like, the collective mode? So the possibility of conformational change. Can they do this just so from sequence in, you know, a protein that's, you know, not homologous to, you know, myosin or something, not homologous to something that we already know has a moving hinge. But
yeah, that's a very good question.
The question of sort of physical approach to kind of mechanics of floppy modes in these structures and sequence.
Right. So as far as I know, I mean, like the main aim is really to predict the fundamental structure, like the fundamental state. And then I would say, for instance, there are other questions in folding that are relevant. For instance, the one which would be how do proteins fold in the cell? Like what's the process of folding? And this one is not really addressed by this kind of methods. The one you were mentioning about the deformations, I mean, yeah, I'm not sure like whether it's, I would say it's, I don't think it's directly addressed by the method, but I think once you have the proposed structure, you could employ some molecular dynamics on it at least, and then find out about soft modes. That's what I would say. However, I mean, it may be incorporated. I have to say I don't really know, but at least I would say once you have a predicted structure, presumably you can use it.
So Genevieve, if you have raised your hand.
I just wanted to slightly turn the conversation with, I think I mentioned this yesterday, but there was a great paper out of Casey Green's lab that looked at the question of dimensionality. And I think a lot of people here have touched on reduced or dimensional reduction tools. And I think this is an area where, you know, theory and statistical physics could really help us in terms of like, you know, they applied a huge number of different machine learning methods, autoencoders, neural nets, and even, basic like PCA, NMF dimension reduction techniques, and basically found that there really was no optimal dimensionality. And the question of, what is the optimal dimensionality in a reduction or in a regression even is like unsolved in math. But there is something to the hierarchy of biology and hypothesis, question specific issues in terms of dimensionality, where I think a focused attention on that area, especially in terms of the work of theorists, could really advance not just the mathematical theory, but also our ability to dissect this data and tune these parameters and find stable and robust metrics. Bin was mentioning in the chat how she builds stability into her models, and that's very important. And I also have found that to be the case, is, stability really breeds robustness. Anyway.
Well,
Yeah.
thank you
Can
very,
I chime in?
I think, yeah.
very much for the comment.
Yeah.
We are getting messages from Janine that we are overstaying our welcome. And so thank you very much, everybody.
Okay,
Yeah.
I'm just gonna say a couple words to conclude the workshop. So I wanted to thank everyone for speaking and discussing and attending, and just emphasize that we really appreciate all the thought and work that in particular the speakers and discussion leaders put into both being visionary and sharing their wisdom. I think a lot of us went out on a limb, talking about our hypotheses and ideas and being provocative, but also we were all, I think, open and receptive to each other's ideas. So I thought it was a great discussion. I personally am a computer scientist trying to learn about both theory and 4D cellular physiology, and I think I learned a lot from these talks. And a goal of this workshop, besides it being a great workshop for all of us that are participating, was to help us plan for the new research direction at Janelia, 4D cellular physiology. And one of the things that I took away from these discussions was that there actually aren't any easy question or easy answers to the questions about what we should do in this new area. There isn't like one model system that we should all be working on, or one type of theory that we definitely need to do, or it's not the case that machine learning will just solve all our problems for us. So instead, we need discussion between people with many different expertise, and this needs to be iterative as we go forward. So from the chat, it seems like many of us learned about fields outside of our own, and there may be even beginnings of some collaborations. And I hope that we can continue these discussions as we develop the 4D cellular physiology research area. Finally, I'd like to thank my co-organizers. It was an absolute pleasure to work with James Fitzgerald, Anne Hermanstad, Michael Reiser, Allison Scrow, and Janine Stevens. And I'd particularly like to thank Janine, who did all the organization of this effort. One of the big draws of Janelia is the ability to organize conferences and workshops like this one with minimal work. And this is largely because of all the real work is done by Janine and Alethea, as well as the IT team. So yeah, I just want to thank everyone again.
Excellent. Thanks, Kristen. I just want to echo Kristen and say thank you to all the speakers, the discussion leaders, of course, the organizers, and all the audience for joining us. As a reminder, the sessions were recorded and they'll be available on Janelia's YouTube channel in the next couple of days. And keep an eye on our website, 40cpjanelia.org, for next steps in our 40CP journey, including recruiting for group leader and senior group leader positions. So thanks, everyone. In terms of the panelists, please don't go too far. I've just sent you a link. We will begin our second closed discussion in about 7 or 8 minutes, around 5 p.m. So we'll see you there. Thanks.